Alright, now we need to look at the second half of Chapter 5, which deals with biotechnology. So first of all, we need a basic definition. What is biotechnology? Biotechnology is the modification of living organisms for practical benefits, usually to benefit humans in some way. Biotechnology is used to produce medications, more nutritious food, reduce our impact on the environment, and also it's used in analyzing forensic evidence. And we'll go through each of these in more detail. Genetic engineering is a much newer concept, and in genetic engineering they actually manipulate the genetic material. So you add, delete, or move different genes around. And you guys are all genetic engineers because you have moved a piece of DNA from a jellyfish into E. coli bacteria to make it fluoresce under UV light. You don't necessarily have to do genetic engineering to get all of these. Some applications you can do that without genetic engineering. Some of them you use genetic engineering to get there as well. So we need to look at some basic tools, and in lab you've used some of these tools already. So restriction enzymes, we've used those in our crime scene experiment to cut up our DNA, and restriction enzymes basically are tiny little molecular scissors. So they cut DNA apart so that way we can isolate individual genes or pieces that we want to look at. Polymerase chain reaction amplifies or copies the DNA, and it's abbreviated as PCR, and this is basically like a copy machine for DNA. It uses changing temperatures to make lots and lots of copies of your DNA. You can use bacteria, inserting DNA into bacteria on plasmids. We did this with our pGlow experiment because we had our pGlow gene. We also had AMP resistance, and it was under control of a sugar. So an on-off switch with arabinose for our pGlow gene. So we could grow them in bacteria using plasmids, and a plasmid is a small circle of DNA that you can add to bacteria and allow them to grow. And then you need to screen or identify which of those growing bacteria actually has the information that you needed. The screen that we used in class was ampicillin, so if the bacteria did not get this little circle of DNA, they didn't survive on the plates because we put ampicillin in there. Here's a picture of a couple of the different tools. So restriction enzymes, they cut DNA, and they cut at very specific locations. So if we have this piece of DNA, every time we cut it with a particular restriction enzyme, it will always cut in the same pattern at the same locations. So that's very useful in manipulating DNA. Amplifying DNA, or copying it using polymerase chain reaction, or PCR, use heat to separate the two sides of the DNA strand. Use enzymes then to copy each of those into a new strand, and then once it's copied, you repeat the process. You heat it up again and copy. In each round of PCR, you double your amount of DNA. That's what makes it possible to take even a single cell from a crime scene and produce enough DNA to analyze, to see who was at the crime scene. And inserting plasmids, so again, a plasmid is just a small circle of DNA, and you can add whatever genes you want. So the gene that you're interested, put it onto that small circle of DNA, put it into bacteria, and it will grow and produce whatever it is that that piece of information codes for, so a protein, an enzyme, things like that. So biotechnology and agriculture, actually breeding crops, so plants and animals, has been used for thousands of years, and that's a type of biotechnology. So you're manipulating a living organism for human benefit. So biotechnology has actually been around for thousands of years. An example of that, something we're familiar with here in Iowa, is corn. And nowadays, corn is this huge ear on the plant. Well, corn is a grass. That's all it is. And the ancestral corn cob looked like that. So substantially smaller, not much to eat, and through just breeding, hybridizing species, picking the plants that we liked the best and planting those seeds the following year, just traditional breeding techniques have given us our massive modern ear of corn in comparison to the original ancestor. Same thing has happened for all domesticated plants and animals. Just breeding and selectively picking which traits has modified them substantially. It's only very recently that we've started using recombinant DNA. And you get the same effect, but much more quickly. So it might take maybe 20 years to breed corn or more, so 20 to 35 years to breed a particular trait or characteristic into the corn. So find a type of corn that gives good yields but is also drought resistant. With recombinant DNA technology, we can cut that time in half. So maybe it will only take 10 years or so to move the DNA and get our corn plant that's drought resistant but also gives good yield. So it about cuts the time in half, but you get the same effect. And the result of recombinant DNA technologies, we have genetically modified organisms, so GMOs, or genetically modified organisms. Some examples of GMOs: golden rice was developed to help especially people in very poor areas of the world where maybe the only food that they have access to is white rice, or people on the edge of starvation. And white rice does not have very many vitamins in it. One it's missing completely is vitamin A. Vitamin A is very important for your eyes to develop and function normally, so if you don't get enough vitamin A, it causes a lot of eye problems, especially in areas of the world where maybe rice is the only thing that they have to eat. They're on the brink of starvation, and if those children don't get enough vitamin A, they go blind by an early age. So by age 6, they go blind because of the lack of vitamin A. So some researchers took regular white rice, and they took a piece of DNA, or a gene from daffodils and a gene from bacteria, and put those into white rice. So now they have golden rice. So the rice looks a little different because it's producing beta-carotene, plenty of beta-carotene. So if those very poor areas grew this rice instead of that, so just switch it out, still requires the same water, land, all of that. So the only difference would be the type of rice that you plant. Then that would prevent anybody in those areas from going blind because it would supplement them with vitamin A. Problem with that was it's not the hungry people that are making those decisions, and governments decided that they didn't want to use that. And so people are still going blind through vitamin A deficiencies, even though there is a fairly easy solution to that problem. We can also reduce our environmental and financial cost of food production. And looking at just the United States, right now, about 86% of all corn planted in the U.S. is genetically modified. 93% of all cotton is genetically modified, and 93% of all corn is genetically modified. And that's based on this year's textbook. However, just the previous edition of this same textbook, so the 2010 edition, we can see that this has shifted. So it went from 45% to 86%. Went from 76% of cotton to 93%, and 85% of soybeans to 93% as well. So the amount of genetically engineered crops is going up. This is beneficial for the environment because if we have genetically modified crops, then we have to use fewer pesticides and other chemicals to grow them. One example of that is insect resistance. So this is a plant that obviously has no insect resistant. So these caterpillars are just eating this entire plant away. This plant is going to have trouble surviving, much less producing a crop. Over here, we've got this worm happily eating all of these tomatoes. Does anybody want to eat this tomato? Would we be okay if our grocery store stocked this tomato? I mean, you could still probably eat this lower half, just cut off all the part that the worm has chewed on. That's not appetizing. That's not going to sell in grocery stores. That's not what people want to have. So the alternative would be, well, the plants themselves can't protect against those insects. So we spray with pesticides and put those chemicals into the environment. Well, then people don't want those chemicals, because if you spray the leaves and our tomatoes with a pesticide to kill the bugs, then those tomatoes could still be covered in the pesticide, or they could have absorbed the pesticide. It could be systemic, and so then it could turn our tomato into poison. It looks nice and pretty, but it's poisonous. So another alternative is to give the plants extra information to make them resistant to the insects themselves. We do that using Bt corn or Bt crystals, and Bt stands for the bacteria that normally is found in the soil. So normally down here in the soil you'd have the bacteria that produces Bt and makes it so that the insects don't eat the bacteria in the soil. All we do is cut out that little piece of gene from this bacteria. So this is where we get our Bt from, and we put that into a plant cell, and then we grow that plant, and now whenever the insect starts chewing on it, it eats those Bt crystals and it kills the insect. So pretty much the first couple bites, the insect dies off, and we don't get this going on with our tomatoes, and it doesn't destroy the leaves. That Bt crystal does not have any problems for humans because of the way our digestive systems work. So we have an acid-based digestive system. So we make acid in our stomach. Well, insects have a basic digestive system, and these Bt crystals only work if they're in a basic environment. So for humans, those crystals just pass right through and never activate because it's an acidic environment. Insects are the opposite, and so it has an effect on them. So it's safe for humans. It's been in the soil for hundreds of thousands of years, so humans have constantly been exposed to Bt crystals, and you're still being exposed to them even if it's not in the plant itself, because it's in the soil and the bacteria that's very, very common in soil. But now we've just moved it to the plant to help us fight the insects without adding all of the strong chemicals that would run off into our water supply or be absorbed into our food and make that toxic. Another benefit, we could add herbicide resistance. So that way we can spray for weeds over our crops and not kill the actual crop, because weeds would use up nutrients and kind of compete with our crops. So that way we're able to grow more crops without worrying about the weeds. That's also a bacterial gene that's transferred in there so that our crops are resistant to herbicide. We can also make them grow faster. So these two, I believe they're salmon, are actually the same age and were raised under the exact same conditions. They're about 18 months old, and this one is substantially larger than that one. The reason for that is they've adjusted the genes in this fish so that it will constantly grow. Otherwise, these salmon have natural growth cycles where they're going to grow for a while, and then they'll just stop growing. And then they'll grow for a while again, and then they'll just stop growing. Well, if we turn off that stop gene here, then they just grow continuously, and we can provide them with the same conditions but get much larger fish. This is beneficial for fish farmers because the same amount of resources, and they produce more food. Our demand for food is increasing, so we need to have increased food availability. So larger fish helps with that. Some problems or concerns that you might have. What about unintended traits? Well, some unintended traits can come about through traditional breeding as well. This chicken over here was produced using traditional breeding. So they were just picking boy-girl chickens and putting them together, and they would pick chickens with the fewest amount of feathers. The idea was, well, if we could get a naked or a featherless chicken, that would be beneficial because a large part of the cost in producing chicken for food markets is getting rid of the feathers. So removing the feathers, separating those from the meat, and not allowing them to contaminate the food. Because really, if you're at the grocery store and you're going to buy a pack of chicken, would you buy a pack of chicken that had feathers in there? Probably not. But this was produced using just traditional breeding, and lots of things have been produced this way. So there are hairless cats and hairless dogs that have been just traditionally bred, so picking parents that have fewer and fewer feathers or less and less hair to get that organism. So that was done by traditional breeding. Some unintended problems with that? Well, it turns out that these chickens are not really economical anyway, because they sunburn very easily without the feathers there to protect them, so you can't have them free range. And also, they're very prone to parasites. The feathers actually help protect against parasites and mosquitoes and other things. So now we've got a chicken very prone to parasites and sunburn, so it needs very specialized environmental conditions, which are too expensive to maintain with large meat-producing farms, so it's much better to remove the feathers later. So that's an unintended trait. You could have unintended traits with transgenic organisms as well. Perhaps if you put a new gene into a plant and it produces a protein, some individuals might have an allergic reaction to that. So an example would be Starlink. Starlink corn was a genetically modified corn that was never meant to be in the food supply. One of the suppliers mixed it up with regular corn, and it was used to produce kind of taco shells that were sold in grocery stores under the Taco Bell label. A lot of people ate those taco shells and had zero problems with it whatsoever. A few people had a mild allergic reaction to that new protein in the Starlink corn, and so they recalled all of those and removed them from the market. Other concerns, some of them are legitimate and rational, and others are not, and we'll talk about those on the next slide. Organisms that may become invincible, so in areas where they grow canola, they've added genes to make them resistant to herbicides. However, those canola plants, if they start growing outside of the farm, maybe in people's gardens, then people can't simply spray and kill them off. They have to actually pull them out, so they become a nuisance weed. Insects could become resistant to Bt, so then those Bt crystals would no longer work, and we'd have to go back to spraying them again. Another argument would be, well, other organisms might be killed by things like the Bt crystals, and that is true. So because monarch butterflies feed on milkweed, which is commonly found next to cornfields, if the pollen from the corn blows over and lands on the milkweed and the caterpillars eat that pollen, they could die from it. However, if we didn't have Bt crystals in the corn crop, we would still have to spray them with chemicals in order to kill off the insects to get a harvest. The chemicals not only kill the insects in the field, those chemicals can blow over if it's even a slightly breezy day, blow over and land on the milkweed and still kill the butterflies, and the rain will wash that chemical off into the water supply, and it can affect humans. And also it's been shown through extensive studies that spraying crop fields to kill off insects also is toxic to birds, and it kills about 65 million birds a year if we're spraying our fields. So it's not a perfect solution, but it's a little bit better than just coating our environment with toxic chemicals. So some legitimate concerns. I mentioned we would get to kind of some legitimate concerns and some not so legitimate concerns. So these are legitimate concerns. Well, is there adequate testing and regulation of these? Well, to give you an example, there's a single strain of soybeans that's herbicide resistant. And to get this one single product out to market, it had to be approved by 31 different regulatory agencies across 17 countries. So 31 separate regulatory agencies in 17 different countries analyzed this soybean, did their own tests and analysis to determine that this was safe to use. Is that enough? Is that okay that we have 31, or do we need more than that? The problem with it right now is we don't know what is adequate. Is that good or not? And who decides that? How do we know for sure with each of these different crops? Eating GMOs could be dangerous. I've already mentioned the Starlink corn. There's also a gene from Brazil nuts that was transferred into soybeans, which would be a problem if you're allergic to tree nuts. If you eat the soybeans, you could have an allergic reaction to them as well. So the problem comes in checking to see exactly what allergy is triggered by these different proteins that are being moved around. And sometimes we don't know. It's very difficult sometimes to pin down exactly what is causing allergic reactions. So that becomes very difficult. And even if we labeled all of them, how do we know? Is everybody going to go and get one of those skin tests? If you've ever had one of those, it's very unpleasant, where they add basically a little patch on your skin and they put just small amounts of different allergens and then wait to see if you have a reaction to any of them. Are we going to do that, have allergy tests for all genetically modified crops, so you can go and get a full-scale allergy test so that way you know what to watch for on the labels? So that's very hard to establish. Loss of genetic diversity, this is a very good concern because our crops are moving genes around. Every plant in a field is genetically identical. They're all clones to each other. And you'll probably see that, especially while crops are starting to come into the field. So if you look at, drive past a cornfield or a soybean field, the corn is all about the exact same height. If you go out there and look more closely, they all have about the exact same number of ears at about the exact same location up the stalk. So it's very, very uniform. The reason behind that uniformity is they're genetically identical. We know from history things like the Irish potato famine. Potatoes, you can cut them and plant the eyes of the potatoes, and they're genetically identical to the parent, and you can do that year after year after year after year and basically get a whole crop of cloned potatoes. Well, that happened in Ireland. There was a particular variety of potato that people liked. It grew very well, and they had that planted pretty much everywhere. Well, a new potato disease came in, got into the country, and got into the field, and the potato crop failed for several years because it was all genetically identical. One plant was susceptible, everything in the field was. There was nothing to prevent that disease from killing off the entire crop. And over 1 million people simply starved to death. That's also why a lot of Irish came to the U.S. to escape that famine. Also, hidden costs might reduce the financial benefit. A lot of companies are producing sterile seeds, and historically, farmers would save their seeds, or some of them, and then replant the next year. However, it takes a lot of money and it takes a lot of time to produce a genetically modified corn. And this argument originally started because companies, they produced a crop that was genetically modified, and they issued a guarantee or a promise. So we guarantee that this is going to perform and do a certain thing. So farmers planted that. So I went to farmers. And that first year it did just fine. Performed okay, and the farmers were quite happy with it. Problem was that farmers were used to saving their seeds. So they saved and replanted. The second year, the crop did not perform the way it had the previous year. Well, they went back and sued the companies for their money because there was a guarantee on that crop. The company said, well, the problem was you can't save the seeds because what's happened from what the company produced up here, that original crop, you have independent assortment, crossover, and random fertilization occurring in your field to your crop, so that mixes up the genetics. So they can't guarantee anything beyond that first generation. And so there was a big lawsuit to the companies won that, and so there's been kind of some stress or strain between farmers and companies ever since. And now, to get around that, companies are producing sterile seeds to make sure that they don't run into this issue again of getting sued for the second years of crops. Now farmers have to just buy seeds every year if they want those particular traits or guarantees in their field. So drought resistance, BT crops, things like that. Here's some less rational fears. One of them that's very common: GMOs are just not natural. They shouldn't be out there in the environment. Well, true. There's a lot of natural things, though, that we still fight against. So having smallpox is natural. It's an organism. It's in the environment. Having smallpox is natural. The vaccine that prevents the disease is a human product. So you could argue smallpox is natural, so we shouldn't allow the vaccine to be used because that's unnatural. It's also not natural to run around attached to a cell phone. That's entirely unnatural. It's not natural to drive a car everywhere. We should just be walking. That's a man-made thing. So this particular idea that GMOs are not natural is less rational. And it's not a valid point for evaluating if the product is good or not, because lots of unnatural things are very common. Wearing glasses and contact lenses to see, having hearing aids, all of those are unnatural, man-made things that are put there to make our lives better and easier. Same thing with genetically modified organisms in our crops. Yes, we've manipulated them, but that's not really a good, valid reason for being against them. With anything, you have to consider the risks and benefits. So genetically modified foods allow us to produce enough food, reduces the cost overall, reduces the environmental impacts and our chemical exposures. So those are all benefits. And yes, it has some downsides. It's not a perfect solution, but it's better than what had been occurring prior to that with all of the pollution.